Vestibular-Evoked Myogenic Potentials, or VEMPs, are audiometric tests designed to evaluate otolith function, which reflects the integrity of the vestibulospinal pathways. These tests are crucial for assessing balance and detecting specific vestibular disorders. VEMPs are categorized into two types based on their recording sites: cervical VEMPs, cVEMPs, and ocular VEMPs, oVEMPs. During a VEMP test, a stimulus such as a tone burst, forehead tap, or auditory clicks is presented to the patient. Short-latency responses are then measured. cVEMPs from the sternocleidomastoid muscle and oVEMPs from the inferior oblique or extraocular muscles. The cVEMP primarily assesses the saccule and the inferior vestibular nerve, while the oVEMP targets the utricle and superior vestibular nerve. This differentiation makes VEMPs instrumental in diagnosing disorders like superior canal dehiscence, where the cVEMP response occurs at lower sound intensities, typically between 60 and 70 decibels, compared to the normal threshold of 80 to 90 decibels. Over time, the clinical applications of VEMPs have expanded. While initially used for diagnosing superior canal dehiscence, VEMPs are now being explored in evaluating other vestibular pathologies, including Meniere's disease. These tests provide a valuable window into otolith function and contribute to a more comprehensive understanding of vestibular disorders. To summarize, VEMPs are non-invasive, stimulus-specific tests that help distinguish between utricular and saccular dysfunction, enhancing diagnostic precision for various vestibular conditions. Their utility continues to evolve as new recording techniques and stimuli are explored.
The cVEMP pathway evaluates the function of the saccule and the inferior vestibular nerve. Here's how it works. A stimulus, such as a sound or a tone burst, is delivered to the ear. The saccule detects this stimulus and sends signals via the inferior vestibular nerve, which is part of the vestibulocochlear nerve. These signals travel to the vestibular nuclei in the brainstem, where they are processed. From here, motor signals are sent down the medial vestibulospinal tract, activating the sternocleidomastoid muscle. The response is measured using electrodes placed over the sternocleidomastoid muscle. This reflex helps us assess saccular function and the integrity of the inferior vestibular nerve. In contrast, the oVEMP pathway targets the utricle and the superior vestibular nerve. Here's the sequence. A stimulus, such as a tone burst or forehead tap, activates the utricle, which detects linear acceleration and gravity. The utricle sends signals through the superior vestibular nerve to the vestibular nuclei. These signals are relayed through the medial longitudinal fasciculus, activating the inferior oblique muscle, PI muscle. The response is recorded using electrodes placed near the labrum. This test assesses utricular function and the integrity of the superior vestibular nerve. To summarize, the cVEMP pathway evaluates saccular function and the inferior vestibular nerve, with responses measured at the sternocleidomastoid muscle. The oVEMP pathway assesses utricle function and the superior vestibular nerve, with responses recorded near the inferior oblique muscle. Both tests are non-invasive and critical in diagnosing conditions like superior canal dehiscence, vestibular neuritis, and Meniere's disease. Together, they provide a comprehensive assessment of otolith function and vestibular nerve integrity.